We've got Dr. Monkol Lek and Katherine Koczwara from the Lek Lab at Yale University. A lot of you are familiar with Monkol and the work that he's done with NDF over these past few years. We're so lucky to have him on our team. And Katherine has been a phenomenal help to him and to all of us. Anyway, they're going to be speaking today about, they're going to give a research update on collected GNEM patient samples. So with that, I will turn it over to Monkol and Katherine, and I will see all of you back at the end of the presentation after Carmen moderates the questions. Thanks so much for being with us, Monkol and Katherine. I will turn it over to you.
So thank you very much, Nancy, for the kind introduction. And for me, it's wonderful to be invited back again to give another presentation. So I'm excited to be here and excited for the patients that are, and families and scientists that are here this afternoon live. And for those that couldn't make it live, that, hopefully you get an opportunity to get to watch this talk and skip to the bits that you're most interested in also. But I will try and keep it all interesting. So I'm very excited to be here, and also excited to be here with another lab member, Katherine, who's a very talented PhD student in my lab, who will talk about some of the research that she's leading, and also the establishment and maintaining a lot of the cell lines that we've gathered from the conferences. So unfortunately, this is an update. We did have plans in 2020 expand it, but then the pandemic hit and things like that. But I'm very positive that going into the future we can expand this in this biobank, and hopefully you can see from this presentation the value of actually doing that, and and hopefully we do cover that and it becomes obvious the value. So the title of today's talk is Research Update on Collected GNE Myopathy Patient Samples. So just as an introduction, who are we? So I'm a scientist at Yale University. I'm funded by the NDF. So we put in 2 grants that were reviewed by the Scientific Advisory Committee and also the scientists that the NDF work with to develop a patient biobank for the GNE research community. And Katherine, as I said, is a PhD student in my lab working on this project. So not just working with the biobank, but working on ways we can design therapies, and also the basic science of some of the questions that we want to ask, but it won't be the focus of that talk, that part of her research. Maybe in a future talk, if people are interested in the basic science, also not just the translation, we can present on that too. So what you'll learn, hopefully, from this talk is what a patient biobank is and what goes into it. So, just demystifying that word, how patient biobanks can further our understanding of the disease itself, how we can use it to discover new biomarkers. In particular, we're mostly interested in molecular biomarkers, or being even more specific, genetic biomarkers. And hopefully you understand the importance of that, and to develop and test new therapies rapidly in using the patient cells, where it's a lot safer. You can't kill the patient cells, we have millions of them. So why are we actually doing it? So just let you know, we're not doing it to satisfy curiosity. I'm a patient myself, so everything we do, even if it is to understand the basic, the basic biology of GNE myopathy, we're always thinking, how can we translate this knowledge? So one of the things we're interested in is actually biomarker discovery, and it's important. It's very important in this context, and I hopefully you see it as a recurring theme from presentation I gave, Angela gave, Kelly gave, and others have given, that you need a biomarker to tell if a therapy is working. So I know sometimes everyone thinks it's obvious, but it's not so obvious, particularly if it's a slowly progressing disease. During the therapy, you may not go from one state to another, and it's not that obvious. And so we're looking for things that are very sensitive to changes, and some of these things are called are more genetic biomarkers. So the RNA. So I'll talk about that in terms of the central dogma of genetics, so you know what I mean when I talk about that. And also we can also use it as a rapid way of testing gene editing approaches, which Katherine's going to touch upon also. So gene editing is still very, very new, and so it requires many, many rounds of optimization, and having a replenishable source of patient mutations patient cells harboring patient mutations, we can rapidly go over all, each one of those iterations and learn how we can do things better and more efficiently. And at the moment, Karen, one thing I do want to say, and I think Stella had highlighted very, very well in her presentation, is that this technology is really, really new. It's not mature yet. So a lot of the things that we're doing with this technology is taking it one step at a time. So using it as ability for its ability to edit the patient cells that we have here, and we'll just confine it to that goal first before we think too far ahead into the future. Because Stella has already highlighted in her presentation very elegantly and well some of the challenges at the moment of translating CRISPR and other gene editing technologies into patients and animal models. So before I start the presentation, just a huge thanks to the team here at Yale that, has worked either directly or indirectly with some of the results that we'll be presenting. So particularly Catherine, who will be presenting the middle part of this presentation. I'm sorry, I forgot to say that. And also Sander, who did a lot of the analysis and processing of the whole genome sequencing data, amongst other things. Our collaborators, Perry and Tarsine, without their help, we wouldn't have had a patient biobank. It was their research protocol and their clinical team that made it possible at these patient conferences to collect patient samples. And also Marianne, just for her support in the design and just consideration of a lot of these things that we're doing. She's just been fantastic help in general on a lot of these things. And, also the NDF, who's funded all this effort. And just a special thanks to Lali. When we came up with this idea, I think it was just 4 weeks before the UCLA conference, patient conference. Most people will go, too hard, well, let's try for next year. But Lali was more like, what can the NDF do to make this happen? And, Lali, Perry, and others at the NDF did make it happen, and it was fantastic. I'm so used to working with people, they'll say, nah, that's too hard, can we just try next time? So it's been fantastic in terms of that. And lastly, not least, the patients and their families, because they're the people that we're collecting these samples from, who, had to go out of their way to actually to give this. And we hope that the research we do is giving back to them. Hopefully you see that in this presentation. So what is a biobank? A biobank is simply a large collection of biological samples used for research purposes in this context. So why establish a biobank? Well, we see it as a way of having a centralized location to store and maintain patient samples. So the things that we have biobanked in the context of GNE myopathy for the NDF is DNA, RNA extracted from biopsies and cell lines, the biopsies themselves before processing. So these are the skin biopsies. We haven't included muscle biopsies at the moment, and cell lines. So the cell lines are again created from the biopsies. So Catherine will go into the details on how the cell lines do get created from the skin biopsies, from the ones taken from the UCLA conference and also the Philadelphia conference. And it also provides researchers an easy way to access available samples. And Catherine will highlight where we have already shared it. And, hope is that if any other researchers are on this call right now or listens to the recording, that we have an opportunity to share the samples with them. And I see it as this: more researchers working on GNE myopathy, the more we'll learn as a team, as a global research team. So what does it look like? So, as a patient myself, I always wonder, how do these things look like? Well, it's not that glamorous. This is what we call a chest freezer that holds all the samples. So it's an easier way to access than our liquid nitrogen tank. It's been a pain in the ass getting things out of the liquid nitrogen tank. So this is the way we're storing the samples. It has plenty of capacity. So from all the cell lines and things that we've actually created, to keep it at the right temperature, it's alarmed and things like that. But we are always looking at ways of creating redundancy, and also a way of, making sure that it's shared and it's not seen as a property of a lab in Yale, but more, the property of NDF. So we'll always be looking at different ways we can actually store it, but that's where it is for now. So before one of the things that I've never ever had an opportunity to touch upon with the patients is just some concepts of collection of patient samples. And I'm only going to briefly touch upon this, and do use this if you're a patient, time to actually read more into some of these concepts. The first is the concept of research protocol. And I'm covering this concept because sometimes patients, one of the great things is patients are very eager to donate, but it's so you understand these concepts. So research protocol is normally a protocol a researcher puts in with their institute. So it outlines the goals of the research, what they're going to do, and also how they're going to protect the patient, minimize their risk, and also protect the patient's privacy, amongst other things. So it's just a detailed document of what you're going to do in terms of research. So anything that involves patient samples will have a research protocol. So unfortunately, sometimes patients do want to give samples, but the doctor they're seeing, things like that, doesn't have a proper research protocol and things like that. And so there are some challenges in collection, and that's why we've collaborated with Tarsine and also with Perry, who have proper research protocols to research into GNE myopathy and also collect patient samples. The next thing is patient consent. So nothing is ever taken without people's consent. So during the consenting process, normally a researcher or a clinician will go over the goals of the research protocol, and also manage expectations. A lot of our research will not directly benefit the patient who is actually donating a sample or donating data or donating information. And so it's also the management of expectations also, and getting them to sign on it so they are aware. Next is patient privacy. So everything that Tarsine and also Perry has collected, the personal information, personal identifying information, we here at Yale do not have access to any of that information. We receive the samples as de-identified. So it's a bunch of letters and numbers and no way to sort of like identify the person. And that way, patient privacy is protected. I don't know who owns what samples, where they're from, and things like that. All I know is they have GNE myopathy, and sometimes I'll have a little bit more information in terms of the age of the person, the sex, and the mutation, and that's all, because that's all the information I probably need to do research. And that way it protects the patients, but it's something for patients to know that, I can't actually say, we've corrected yourselves in the research setting, things like that, because I don't know who owns what, and it's just that way to protect everyone's privacy. So before I touch upon some of the biomarkers, I'll just touch upon the central dogma of genetics. So you start off with DNA. Every cell has all the DNA, so that's instructions to make everything the cell needs to make. So all instructions to make you. So all the DNA is called the genome, and there's two sets of 22 chromosomes, and then there's the sex chromosomes. And with that, RNA is being made, so it's a kind of a copy of the DNA, and from that, proteins gets made from that. So it has instructions to make protein. So we have DNA, RNA, and protein. A lot of this talk will cover the DNA and the RNA. So one of the things we did collect was blood. At the moment, when using the blood just as a means to get high quality and large amounts of DNA. So we've extracted that from 40 blood samples that were collected over the two conferences. At the moment, there is nothing in particular that they're sort of like destined for, but a lot of them will be used for whole genome sequencing later on. So this is an effort largely performed by PerkinElmer, as the NDF has the contract to do 100 genome sequencing samples. And so saliva was collected at patient conferences. So in India, Israel, and also at the Philadelphia conference. And from those samples, we actually performed whole genome sequencing. Well, PerkinElmer did, and made the data available. So the goals of genome sequencing, so this is sequencing all the DNA, in a person is to characterize the genetics of GNE myopathy. One thing I'm going to emphasize is the genome sequencing effort is not an effort to find what is causing your disease. Every patient that was enrolled in this study, from my understanding with PerkinElmer, already has a definitive genetic diagnosis that they have GNE myopathy. So it's more to characterize the genetics, not to actually find a genetic diagnosis. And also, just a reminder, this is in a research setting, so we don't have the ability to directly report any of these findings back to the patient. And as I had already touched upon, these are de-identified samples, so we don't know who owns them. Only PerkinElmer, who have the research protocol, do know. So we sequence families with different mutations, including non-manifesting carriers. So the hope is that maybe the non-manifesting carriers will actually tell us a little bit more about the genetics of GNE myopathy, if they do indeed exist, and they're validated. And lastly is to identify additional potential genetic contributors to the disease. So are there other variants across the genome, across, say, for example, not other non-muscle disease genes that are making the disease more severe or less severe? So this is a very sort of like open hypothesis. And my understanding is, from my experience, is that we may not be well powered at the moment to actually ask that question in a very statistically rigorous way, but it's something we have to get a start on. And the thought is, if there are genetic modifiers, as we accumulate data, it will become, there may be a threshold of statistical significance that we can conclude. So we've done genome sequencing of 40 individuals across 28 families. So in the samples we collected, I prioritized it with PerkinElmer to do all affected samples and family members if we have both parents. So we'll do both parents and the affected. So in genetics, we call that a trio. So we've done 21 families, so 21 trios where we have both parents and the affected. And so the remaining, if you can crunch the numbers, will just be just the affected individuals. And so there's 21 families with 2 pathogenic variants in GNE. You can see this variant here, also known as the Persian Jewish mutation, is probably one of the most, this one's I think the South Asian mutation. This is the Persian Jewish mutation. You can see we have some that are quite common in that. So the analysis is ongoing. I'm not going to go into the deep, weeds of analysis. We have sort of like planned for the future. And also there will be another wave of sequencing to hit the 100 based on samples we've collected from blood, samples that we have already collected through a collaborator in UCLA, and also other collaborators that may contribute for the next wave, using the information that we've learned from this wave to pick the next best possible set of the remaining 60 to be sequenced. So with that, I'm actually going to transition to Katherine to talk about establishing fibroblast cells from skin biopsy.
Hello everyone, and thank you for giving me the opportunity to speak today. As Monkol mentioned, I'm going to talk a bit about how we actually go from collecting the skin biopsy to being able to perform research on cells extracted from that biopsy. So the first step in this process is to take the skin biopsy and get it shipped to Yale. So this was done by our 2 clinical collaborators, Perry and Tasneem. And so here's an image that shows the layer of a cross-section of the skin. And so as you can see, there's 3 layers: the epidermis, the dermis, and the hypodermis. And the cells that we want to extract, the fibroblasts, are located in the dermis, so in this layer of the skin. So a small punch is taken that makes sure to get into the dermal layer that contains the fibroblasts that we want to extract. So from the initial skin biopsy, sort of the first step in this is to sort of extract that epidermis layer that we're looking to extract the fibroblasts for. So that's done in a series of steps. So first, the samples are received at our lab at Yale, again as sort of de-identified samples. And from this, sort of any adipose or the other layers of the biopsy that are not in the layer that we want are removed, and the tissue is processed into smaller pieces. So once that's done, we can go about actually extracting the specific cell type that we're looking for, the fibroblasts, from the sample, again in a series of steps. So the first step is sort of digesting all of the extracellular matrix or things around the cell type that we're looking for. And this allows us to then spin down the sample and collect the cells, which are very hard to see. They're just in a tiny pellet at the bottom of this tube, so barely visible to the eye. We can then place these cells in a flask where they'll then grow out for about a week or so. So as you can see here on the first day after the cells are placed in the flask to grow out, on the first day you cannot really see anything when looking under a microscope. But by day 4, you're able to see some of the fibroblasts growing attached to the bottom of the flask. And after about a week, so here at day 8, you can see that the fibroblast cells have expanded and filled the bottom of the plate. And actually in this image, you can see them growing out from a tiny portion of the skin biopsy. So once we have the fibroblasts growing, it's really important that we're able to continue to use them for a long time in the lab. So in order to do this, we introduced an H-TERT agent that basically just allows the cells to continue to grow in the lab and allows us to share them with other researchers who can also use them in their lab without the cells dying. And once we've performed this process in the cells, we can then place them in tubes that then can be stored in liquid nitrogen or the cryofreezer that Monkol showed a picture of earlier. Given that GNE myopathy is a disease that primarily affects the muscle tissue, we want to be able to transition these skin cells, these fibroblasts, into a more muscle-like cell lineage. So in order to do that, we have them express factors that are present in muscle cells but not fibroblasts. So then in that way, we can go from a fibroblast cell into the myoblast muscle cell, which we can then differentiate into myotubes. So here you can see when, again, looking under a microscope, you can see that the shape of the cells actually changes when we do this. So it goes from these fibroblast cells to when we differentiate them into myotubes, you can see this sort of like long, more muscle-like myotube that's forming. So again, mimicking a muscle cell type. And then we can also confirm this by looking into sort of factors that are expressed in muscle cells, but not in skin cells or fibroblasts. So as you can see, we're looking at 2 different factors in our cell samples. One is myosin heavy chain and the other is dystrophin, both of which are expressed in muscle cells. So we can see that the dystrophin in green and the myosin heavy chain in red are found only in our induced myotubes or our muscle-like cell, but not in our fibroblasts. So this just shows that we are able to successfully convert these fibroblast skin cells into myotubes. So this is a sort of a summary table of all the samples and cell lines that we've collected and where each sample is in sort of this process of going from skin biopsy sample collection to conversion into myotubes. So green is steps that have been completed, yellow is in progress, and black is steps that are applicable for that particular sample. So what I want you to appreciate is this has been a pretty extensive effort with a lot of cell lines. We have 19 GNE myopathy patient samples and 14 unaffected control samples that were gathered from unaffected family members of the GNE myopathy patients. And for each of those cell lines, we have the primary fibroblasts, the immortalized fibroblasts, the myoblasts, which we can then differentiate into myotubes. So to date, we have a total of 96 cell lines created, which is, great to see the biobank expanding to such a large number. So in addition, one of the main goals of establishing this biobank was to share it with other researchers. So here's just a summary of, some of the different researchers we've shared this cell line to. So Noah at Ohio State, Kelly at Mount St. Joseph's, Marianne, and who works closely with Dirk. So luckily, we've been able to sort of utilize this biobank and share it with other researchers so they too can work on these cell lines that we've collected. So that was a bit about how the biobank is established, and now I'm going to talk a bit about what research we're doing in the lab here utilizing the cell lines from the biobank. So as Monkol mentioned, one of the main goals of this project was the determination of GNE biomarkers. So there has been a few previous studies, including the one pictured here from Marianne's group, that has looked into sort of SNA lectins as a potential biomarker. But we're really interested in looking at RNA as a potential biomarker. So our goal for this is to perform RNA sequencing of the GNE Myopathy biobank. And so we're looking to do this on all of the sample types that we have. So skin biopsies, the fibroblasts, the myoblasts, and the differentiated myotubes. And when we perform RNA sequencing, so looking at all of the RNA that's present in each of these cell types from both affected GNE myopathy patients and the unaffected control samples, we then look to see which genes or which RNA is different between the affected and the unaffected samples. And then this will hopefully also allow us to look at sort of which pathways, which biological processes are also different between the affected patients and the unaffected control samples. And these, which genes could be differentially expressed or which pathways are affected, could give great insights into biomarkers to sort of determine any potential efficacy of any potential treatments. So currently we've sent RNA from 17 skin biopsies and 33 cell lines for this RNA sequencing, which some of the preliminary data we recently received. So this was some preliminary data from the 17 skin biopsies that we sent for RNA sequencing. And one of the things that postdoc Nicole in the lab looked at is sort of the quality of the RNA that we were able to extract from the skin biopsies. So just looking to see how it compared to publicly available RNA sequencing data sets. So what we saw was that our biobank skin biopsy RNA samples in pink clustered very nicely with other RNA sequencing experience on GTEx of skin biopsies and not with some of the other skin types, which is a great indication that our RNA extraction was clean and the sample processing was done correctly. And then also another postdoc, Sander, in the lab looked at sort of the clustering and gene expression analysis of our affected samples in blue and the unaffected samples in orange. And what you can see is we were sort of seeing a separation in clustering, so there were differences between the affected and unaffected samples. And this is something that we're looking to investigate further. So one sort of thing that is difficult to overcome with RNA sequencing is that even though you're comparing unaffected and affected samples, they're not genetically identical. So even parent samples, which were our unaffected control samples, have only share about 50% of the DNA with the affected individuals. So in order to minimize that genetic variability that could affect sort of this ability to detect biomarkers, one of the other goals that I'm working on is the creation of isogenic cell lines through prime editing. So an isogenic cell line is essentially a cell line that where the only difference is the presence of the GNE myopathy causing variant or not. So the rest of the genome is identical because they're derived from the same cell line. But when the prime editing process occurs, it will correct this GNE myopathy causing mutation into the wild type. And the way this is done is through a relatively recent genome editing CRISPR tool called prime editing. And the way this works is that there's the prime editor is composed of the CRISPR-Cas9 nickase fused to a reverse transcriptase. And then this is combined with the prime editing guide RNA, which contains the guide RNA target sequence, the scaffold that binds to the prime editor protein, the template of the genomic edit that you want to occur, and then a binding sequence that can bind to the DNA. So this Cas9 protein and the guide RNA will form a complex, which can then target the specific place in the genome, in this case, the GNE gene that we want to edit. So the Cas9 nickase will cut one strand of the DNA, at which point it will open up, and the reverse transcriptase can then newly synthesize the genomic edit that you want to make using this template RNA. And this results in, when the DNA closes and the nick is no longer present, in the edited strand that has the wild type GNE mutation that we've changed, and the mutation strand is cleaved off. So in the lab, the first thing that I wanted to do was test our ability to do this prime editing using a control region. So these plots are Sanger sequencing, so just showing the DNA, the genomic sequence of a very small region that I was looking to perform prime editing in. So you can see on the left is the untreated sample, so very clean sequencing. But then in the right, when I apply my prime editing construct, which was highlighted in blue, was the area that I was trying to change or correct. And I was able to show that at about 70%, I was able to achieve the desired edit of CTT, was the genome edit that was occurring in this instance. So after I had established the technique in the lab, I then went to show that I could edit within the GNE gene. So just in a HEC control cell line that we have in the lab, I then looked to target the genomic loci of the M743T GNE Myopathy mutation. So just instead of going from the mutation to corrected, just to target the loci going to create the mutation at this locus. So I was able to achieve, let's see, there we go, achieve this edit at about 11%. Which is what you can see here. So once I had shown this editing in a control cell line, my future plans for this gene editing project would be then to test this prime editing construct in the patient cell lines to actually correct the mutation. And this will allow me to create these isogenic cell lines with the corrected mutation, which we can then use to send for RNA sequencing. And then once this has been performed, I'll also look to design and utilize prime editing constructs that will correct the other mutations that we have in the GNE biobank. So with that, I would like to thank everyone in the Lek Lab, as well as our collaborators, Perry, Tarsine, and Marian, as well as the NDF for funding a lot of the work of this biobank. And I think, Monkol, I will give the screen back to you.
Great. Thanks, Catherine. So I'm going to switch, and I thought to present this to end it off. Okay, this is not GNE myopathy, but it's putting it all together, like what our lab and other labs, a lot of the labs that the NDF collaborate with could do if they had all these things. So and of course the planets align. So you can see all, I think, 5 planets aligning in this picture. If we had patient samples, a great biomarker, and and a therapy to develop, and also, as I think Stella had presented, a great mouse model. So I'm going to present on that as sort of like an aspiration of where NDF and the scientists at NDF are moving towards, and have actually iterated on this. But it's a hope that we can iterate again now, powered by patient samples and all the other things that we know. And that includes also data too, in terms of natural history and things like that. So I'm not the best to talk about the long list of things of putting it together, but this is just something that our lab has worked on in the space of another neuromuscular disease, Duchenne muscular dystrophy. So I'm going to give you a case study of a patient that had a genetic report, and based on that genetic report, we had an idea. And the first thing is implementing that idea actually required us to actually perform whole genome sequencing, because at the moment a genetic report is only gathers enough information to make a genetic diagnosis. It's not actually there to sort of like springboard towards a customized therapy, which is what we wanted to do in this scenario. So we actually perform whole genome sequencing. So like with the GNE myopathy studies that we're doing, we perform whole genome sequencing. And for those that haven't seen next generation sequencing being performed for whole genome sequencing, what you're seeing here, all these little rectangles here are little sequencing reads and where they map to the human genome. So in this case, they map to chromosome X, and here I'm just showing around exon 1 of the DMD gene as the region that it's actually mapping. So this exon here of the muscle exon. And you can see here that we have no sequencing reads mapping here, and that represents a deletion in the patient. So a part of the genome that's actually removed and not present, and that takes away exon 1, the person's muscle exon 1, and also the promoter region. So it's unable to switch on the muscle version of that gene. And if you look hard enough, the mother, you can see this represents the coverage. You can see half levels of coverage. And the reason why you see half levels of coverage is the mother is a carrier of this deletion on one copy of her X chromosome. And then the affected is a male patient and only has one copy of X and therefore has a deletion there. And what we can do with whole genome sequencing is map the exact place the deletion starts and where the deletion ends, or the left side or the right side of that deletion, which I'm showing here. I won't go into the details, sequencing details. The other thing we perform, like in GNE, working on the GNE myopathy patient cells, is we perform RNA sequencing. Instead of a skin biopsy, we did it actually on a muscle biopsy. And what the muscle biopsy showed us is it gave us clues. So here is the muscle exon 1. And these arcs represent sequencing reads that span from one exon to another exon. And you can see here, this is the cortical isoform where a read spans from this point to this point here. And so what we believe is happening is, although this patient can't express their muscle isoform of the dystrophin gene or the DMD gene, they can actually express the cortical isoform. So typically found in the brain, but they've actually switched it on in their skeletal muscle or regular muscles that we use every day. And what that means is, well, one thing is that the only difference between the cortical form of this gene and the muscle form is just a few amino acids at the start. So we know that it probably has therapeutic benefits. And we believe this is the reason why we still see some dystrophin protein in this patient. As you can see here, a dilution series to a patient, and here is the muscle biopsy of the patient and stain. The green represents dystrophin. Here it's normal. You can see it perfectly lines the cross section of a muscle and the muscle membrane, and here you can see some patchy staining. So we believe it's actually the switching on of this cortical isoform that is actually rescuing the patient's phenotype. And what that means, okay, in terms of biomarker, is the cortical isoform is a potential biomarker that we can use, because the idea is the patient already does some compensation and slows the progression of the disease. Can we create a therapy that actually can improve upon what the patient is already doing? And if we know what we're targeting, then we know what we're measuring. So this is the biomarker in this case, which is we can use the RNA or we can use the protein. So in this presentation, I'll only show the RNA. So we actually designed a custom CRISPR-based therapy, which goes to that place of the genome that I showed you and switches on the cortical isoform and makes it express itself in the skeletal muscle. So I won't go into too much details. We showed a proof concept in cell lines. So this is in patient cells, just a cell line that was easy to work with. I think in our previous presentation, Angela or I was talking about the value of using cell lines as a way of quickly validating something because it's easier to work with, but ultimately we want to work with some patient cells to see, does it work in that context too? And we're able to switch on all three isoforms, the muscle, cortical, and Purkinje isoform of the DMD gene. Then using the patient cell, we're able to also upregulate the cortical isoform and also show the protein. So it's this band here, and also show the protein restoration, which is colored in green, the dystrophin protein in the patient cells. So what we're most interested in is now, can we actually test this therapy in an animal model? And the great thing about DMD is that it has a great animal model, and not only that, it has what we call a transgenic model with the human copy of the DMD gene that we can actually target and switch on. So use the same therapy that we tried on the patient cells on the mouse model. And here's some early results showing that it indeed does work on the mouse model when we use an AAV to deliver, so adeno-associated virus, the serotype 9, to actually deliver it to skeletal muscle and also cardiac muscle. So here you can see lanes 1 and 2 are the untreated mice. Lanes 3 to 6 are the ones that received a low dose. Lanes 7 to 10 are the ones that received a high dose. And this is at the 4-week time point. We just received the muscles for the 8-week time point. So this band here represents the biomarker. So in this case, the cortical isoform, the DMD gene, we want to upregulate. So the point I'm trying to make here is we had the DNA of the patient, we performed whole genome sequencing, we had a biopsy, we performed RNA sequencing, and from that RNA sequencing, it gave us clues for what would be a good genetic biomarker. And then using that knowledge, we designed the therapy and then validated the biomarker and the upregulation of the biomarker. So this is how patient cells and also having patient data can actually inform us in developing therapies, discovering biomarkers. So I just wanted to present on that example. It's not GNE myopathy, but it's another project going through our lab. So I just want to end with the highlights so far with the NDF Biobank, the NDF funded biobank for GNE myopathy. So as what Katherine said, we have 96 cell lines, 33 primary fibroblasts that have been immortalized. So that just means that they can continually divide and we can renew it and share it with more researchers. And also MyD converted fibroblasts. So they have a more muscle-like character that we can also create those myotubes that Katherine showed in her presentation. In terms of data generated, so genome sequencing of 40 samples, a lot of that was driven by PerkinElmer, but we helped in terms of the selection of the samples and coordinating, and we're working with them to coordinate the next wave of sequencing, whole genome sequencing. We've done RNA sequencing on 17 skin biopsies and 33 different cell lines. So that's 11 fibroblasts, 11 myoblasts, and 11 myotubes from the same patient, from the same donor. So it's a way of saying what information can we get from fibroblasts compared to myoblasts compared to myotubes? Because one of the concerns is at the moment our expression data is from skin biopsy, and patients are probably smart enough to think, well, my muscles are affected. Why are you looking at my skin? Well, one, gene is highly expressed in the skin, but two, this actually tries to give us a little bit of a sneak peek using the skin biopsy to see how it would look like in a muscle-like cell line. But I must stress a muscle-like cell line. We're also doing proteomics. So we want to understand what proteins express in one of the biobank cells if we did proteomics on the fibroblasts, the myoblasts, and myotubes to ask the question of how does that protein environment change? And if we go through, if we've gone through the data deep enough, if we've generated enough data, we may be able to see post-translational modifications, which may have more of an impact on the protein level compared to the RNA level. So I'm going to end off with the key takeaways. So hopefully some of this you can instill. So developing patient biobanks take time. So all the good biobanks, disease biobanks that I've worked with takes time. It's not done in 1 year or 2 years. It's, this sort of like this constant ongoing effort over years, but it is extremely valuable resource once it's actually established. But it's always having a long-term view instead of like, what will the biobank give us back today? I don't think, it will give us, everything we want today, but it's to think in terms of the long term. And, I'm really grateful that NDF and the leadership in the NDF see it that way too. Biobanks allow for the discovery of genetic biomarkers. Hopefully, Catherine has shown you, looking at expression patterns, you already can cluster the affected versus the unaffected. And as sample sizes grow, our ability to cluster and differentiate will be even better because it'll be bigger data. So, it's not perfect, the figure that she showed, but I think it will get better with more data. We can also rapidly test different therapeutic approaches. So Catherine has just shown something in terms of gene editing, and sort of like her plans to use it for testing different gene editing approaches. And our collaborators are also trying different things in terms of therapeutic approaches or just understanding the disease better or discovery of biomarkers. And the last thing I showed in the example of another project our lab's doing is that patient samples can definitely drive the development of customized therapy. So understanding what's causing the disease, the genetics of it, we can actually design something very customized either to that mutation or to that class of mutations that affect that gene once we know the function of that gene and where it's actually needed and the levels that it's needed at. And with that, I'd like to thank everyone for their attention, giving up their Friday afternoon, Friday noon, or wherever you are at the world. And if you're listening on the recording, just giving up your time, just to skip to the bits that you think are the most exciting.
Thank you. Thank you, Monkol. Thank you, Catherine. I think that was a really interesting presentation, and I think you've given everybody a lot to think about. And I think, Monkol, the context that you put things in with the example even of a different disease really helps us understand why you're doing what you're doing and where we hope to get with it. So with that, I will introduce Dr. Carmen Bertoni and let her go ahead and answer all of your questions. I'll be back just at the end of the Q&A for 1 minute, so please stay tuned and feel free if you have any other questions. I see there definitely is a queue of questions in the chat line, but if you have any more questions, go ahead and add those and Carmen will integrate them into the conversation. Oh. Carmen, you're muted. We can't hear you, Carmen.
Guys, I do this all the time. I'm so sorry. Eventually, by the end of this series of seminar, we'll learn to switch on the mic when it's my turn. I wanted to just say, I was just saying that I was thanking Monkol for the presentation and for the insight that he's been able to share with us. And I'm going to go straight to the question. The majority are for Monkol. And I'm going to go straight to Mangesh's question, which I think is what everybody wants to know. Are there any findings from the genetic analysis of patient and family samples that were surprising and that you can share with us?
Well, first of all, that's a great question. And, just reflecting upon the questions that we want to ask. So one thing that I want to reiterate that the genetic studies was not to find a genetic cause. So, the inclusion criteria that PerkinElmer had and that the NDF had was that the person already had a genetic diagnosis of NDF, of GNE. And so any genetic findings we'll find, we'll be looking for either genetic modifiers or something, strange, okay, that we would see in whole genome sequencing. The only thing that whole genome sequencing adds value compared to, say, panel-based sequencing and exome sequencing that people would have received if it was a recent diagnosis is non-coding regions. Our ability to interpret non-coding regions is still very, very rudimentary. So that won't give us any extra information. What it may give us is rare variants that are actually harbored in other muscle genes or muscle disease genes or genes related in the sialic acid biosynthesis pathway or a related pathway. Unfortunately, those things actually require power to, you can make these observations, but you have to make the observation of how often would we see this observation by chance. And to increase that statistical significance, unfortunately, 40 samples would not be enough to answer some of those questions. But the thing is, it is ongoing. So some things need to be powered by larger amounts of patient samples and some things don't need to be. But in terms of finding something surprising, the short answer is no, but it's the hope that when we do sequence some of the samples collected by our UCLA collaborator, that particularly the ones that are deemed, big emphasis on deemed non-manifesting carriers, we may see something there. But, I don't think we will, but they are the most interesting. So the most interesting samples, if people are aware for the next wave of 60, are the ones that are unusual, because the unusual may manifest something genetically. So the 40 samples we're done at the moment, I wouldn't say any of them were unusual. So, and therefore we're unlikely to find anything unusual in their genetics. And if we were to, it's very subtle and will require a lot more data.
Can you expand a little bit on the unusual samples? In other words, we have seen from your presentation that you have quite a few samples from a certain type of mutation. When you say unusual sample, are you referring to additional type of mutations, or are you referring to differences in phenotype, for instance? And can you elaborate a little bit on that?
That's a great question for me to elaborate on. So when I talk about unusual, in this context, I'm more thinking about in terms of phenotype. So those that are either non-manifesting or manifesting later on in life, is there a genetic contribution to that unusual phenotype? Or those that are getting GNE myopathy very, very early in life, could there be an obvious genetic modifier? But everything needs to be validated, either experimentally or with more samples that carry that genetic modifier or modified gene. But touching upon the other thing that you mentioned, Carmen, one of the things we do notice, because the collections were in Israel, United States, and also India, that there are some mutations that are not represented at the moment, and that's why we rely on, say, our good collaborators in Japan, Nishino, to make sure that we have good coverage, to make sure that we haven't left any stone unturned. By only sequencing people from particular regions in the world and probably particular mutations, we miss out on that opportunity.
Okay, and from what you're mentioning before, we have seen that you're collecting sample from patients as well as the relatives. Can you please explain us why it's important to include the relatives also in the collection of samples?
So so one of the reasons is traditionally we've used the relative as an unaffected control. So it's not the best control, as Katherine has said in her talk, but traditionally the parents have been used, or the sibling has been used as an unaffected control, because genetics would say that they share 50% of their genetics with a family member, normally a parent or thing. And that's why it's important. Another way it's important when we do whole genome sequencing is we can look at what we call segregation or transmission of variants. So if we have a hypothesis, we can look at the transmission. So one of the hypotheses in a lot of genetic studies I'm involved in involving parents and the affected child, is looking at de novo variants. So variants that are only created, new in the next generation. So neither of the parents carry that variant, and it was created in either the egg or the sperm and passed over. But, this is not the inheritance mode of GNE myopathy, but it's just an example of why you would need parents in a genetic study, and why you would need parents, say, in a study where we're collecting the cells.
Okay, so you would say that the collecting the relatives is still an important stage, even though science now is moving toward gene editing technologies as well as a mean of using it.
Yeah, but the family member also can, in some cases, we know that the heterozygous or the carrier doesn't have a phenotype, but if someone looks very carefully in some assays, we may see intermediate levels that aren't disease causing, but may be interesting if you want to study the basic biology of it.
We have a comment from Tara followed by a question. And Tara is saying, I like the fact that skin samples, the procedure was less painful than a muscle biopsy. Would you say that you're able to get the same information from a skin fibroblast compared to a muscle fibroblast? If so, which is more applicable for a successful GNE biomarker?
That is a fantastic question. I would say it's not as good. I know Marion and other researchers on the call, and if they disagree, do chime in on this. It's not as good as a muscle biopsy, but when you're at a conference, you get what you get, and as Tara has mentioned, it is less painful, it's less invasive. It's not as good, but what we could do is, it's sort of like a balance between, or a compromise between patient suffering, because like Tara, I've given a muscle biopsy before, and excuse my language, it bloody hurt. And I do not want to give a muscle biopsy again. Okay, and I've given a skin biopsy, I didn't even notice that happened. I put a band-aid on my leg. I have a bit of a scar, it's wrecked my leg, but other than that, I'm fine. So as Catherine is saying, we convert the fibroblasts into myoblasts, but I would say they're myoblasts-like. But it's a good compromise, and it's a good substitute, and that's why Catherine has actually done RNA sequencing, taking 11 donors and saying, what do we see at fibroblasts, myoblasts, and myotubes, and then compare it on what we typically would see in true myoblasts and true myotubes. And and some of these patients have been donating before. Thank you very much, patients on the call. And for those that have donated to other research studies, maybe we can get the true myoblast and then compare again on the RNA-seq and then ask that question. But the last part of the question that Tara asked is, one of the reasons why I think that this is sort of like a thing to think about in terms of feasibility is, if you're going to go through a clinical trial, I would rather give 3 skin biopsies than 3 muscle biopsies, say, during a clinical trial. So it's the hope that there are some biomarkers that we do see in skin, which is less invasive, and that does replicate in true myoblasts or muscle biopsies. But we can use our myoblast-like cells to say what is the likelihood. So hopefully I tried my best to answer that question, Carmen.
Yes, I think so. So in other words, what you're saying is that using the skin biopsy, we are likely, at least in the future, to reduce the number of muscle biopsy that will have to be taken to verify the efficacy of a therapy. Is that correct?
Yeah,
so
but the answer will always be the muscle biopsy and the muscle cells, but I think this is sort of like intermediate, a good compromise.
I agree. So Mangesh is asking a question concerning the CRISPR technology. Can you please clarify or talk in more detail about the success of edits, particularly the 70% versus 11% that Catherine mentioned on her talk?
I'm going to let Catherine answer some things because I need a break. Catherine, can you answer it?
Yeah, so the 70% that we were seeing is sort of in a construct that the lab that published this prime editing technique had optimized. And the 11% is what I was able to get using the constructs that I designed specifically for GNE myopathy. So as I continue to work on this project, one of the goals is to work on sort of designing the prime editing constructs to, increase the efficiency from that 11% higher is sort of a continuous goal when working on CRISPR editing. But the fact that it is 11%, although it might seem not a lot, the fact that it's detectable is something that's a really good sign that I'll be able to optimize it further and increase that efficiency.
Okay, that sounds great. Thank you so much, Katherine. And
I just wanted to come in, if you don't mind, I'll just chime in. So just to reiterate what Catherine's just saying, that this is just the first attempt. So, and we, Carmen, as a scientist, that it's every time you try again, it's about trying to improve it. So this is just, very early preliminary attempts. And also one recurring theme is the importance of patient cells. If we didn't have patient cells, we wouldn't be able to reiterate on these attempts, because we don't want to edit cells that have no relevance, basically.
I'm going to ask a question on my own at this point, given my background on gene editing. So already the 11% is a pretty good number. And as you mentioned, Katherine, your main goal here is to try to do the RNA sequencing and the genomic sequencing from corrected, what we call isogenic. So the same cells from the same patient, but before and after the correction. So my question is going to be, how much more do you need to be able to achieve in terms of efficacy of gene editing to be able to isolate a pure population of cells that are 100% corrected? I'm not saying you can isolate corrected cells, but clearly 11% is going to be more difficult. What would say is you have to achieve to be able to purify a sufficient number of cells to do the pre and post analysis?
Well, before Katherine answers that, that's a great question, because Katherine actually did some screening. She has some, some roundabout numbers of how many she needed to screen.
Yeah, so in my initial screening with that 11%, I was able to get a lot of heterozygous cell lines, screening about 25 different colonies or clones of single cells. So I anticipate that I'll need to either screen a much larger number of cells or try to increase the efficiency in order to get a homozygous corrected cell line.
Okay, thank you. And I think we, if I'm not mistaken, we are done with the question. We are in perfect timing within the 1 hour. So I want to thank everybody. Thank you, Katherine. That was a great presentation. I know how difficult it is when you are a graduate student to give presentation, but you did an excellent job. And thank you, Monkol.
Yeah.
No worries. It was great to share the afternoon with everyone.
Thank you guys so much.
For having me.
Thank you, Carmen, also for your help in all of this and in helping us tie all of the science that we're working on together, because as Monkol mentioned, this international team of scientists that we have working on this disease has just grown so much over the last couple of years, and we're so lucky to have such esteemed people working on our cause. So with that, I really just want to thank especially
actually, Nancy, if you don't mind, and we can get on the line for it, I have one last question that it keeps coming up at the NDF, the SAC committee, and is Monkol, what are the patients that you are really missing right now in your pool that we as an NDF can help you recruit?
That's a fantastic question. Thanks for remembering, Carmen. I totally forgot. I thought I was free to go. So
yeah, me too. Sorry, I should have asked before, but
I'm just kidding. So for the patients listening live, and for patients and clinicians that are listening on the recording, one of the things that we find in terms of Katherine's at 11%, she's optimizing. I'm not going to go on the technical details of it, but the best set of mutations to work on, this is not the saying we're not going to work on all of them, but the best set are the homozygous ones. And you get homozygous mutations when they're the common mutations, and it's just by a chance thing that if they're common. So we have some homozygous of the Persian Jewish mutation, but we don't have some in terms of cell lines. In terms of DNA, we do have good representation of some of these in terms of homozygous form, but of the South Asian, the East Asian, and I think the Northern European. So we have a lot of them in compound heterozygous state, but not homozygous. Homozygous are much easier to validate our editing efficiency. As I said, I'm not going into technical details, but it's just easier to validate. If it's easier to validate, then it's easier for Katherine to optimize to know if she's getting better and better at doing it. Otherwise, at heterozygous state, it's almost impossible to work with in terms of editing.
Thank you. Thank you, Monkol.
Thanks, Carmen, for bringing that up. So to wrap it up, I do want to thank everyone for joining us. I want to remind everyone that on our website, curehibm.org, you can find the recording of this shortly and the recording of all of the past speaker series events that we've had. So with that, I thank you all for your time, your energy, and your collaboration. And if you have any questions or comments, as always, you can reach out to any of us at info@curehibm.org. I hope that you all stay safe and you all stay healthy and you all stay in touch. Thanks again, Katherine, Monkol, and Carmen.